no good videos on substrate level phosphorylation. Ugh, they're terrible. So that took 15 minutes. I meant for it to take less, but I'm glad it took what it took because there's nothing for me to show you that's any good there with the video. So we did just this part before. Now, although this is just saying glycolysis, we're going to go through everything. We're going to draw it all. We're not going to learn it all, but we're going to draw it all one time. The reason we're going to draw it all is because we're going to have our drawing in the top half, and then we're going to learn about it and write about it underneath. So, for those of you who like to squish everything on one page, this is not the time to do that. This is the time to just do what I do. So then we'll have the rest of this page. So that looks like this. And then we'll finish with the rest of what happens in this same mitochondrion with all of this down here to write. So this is about drawing. This is not about learning yet. Okay. So here's what I'm going to do. I'm going to start off with my page here. I'm going to help myself remember that I'm not allowed to draw below, I don't know, that's probably, what, 2/3 maybe, up there. This is all I got. So, I'm going to start off with glucose. Now, again, let me move my head. This is not meant to be exact or perfect. This is just to help us get a feel for the level of detail that we need to know what's going on. So over here, I'm going to start with glucose, and I'm going to represent that as this just 6-sided molecule. Just because it has 6 carbons, I'm not saying that's what it looks like, but that's going to be my visual representation. And I'm going to let myself know that's glucose. And I'm going to remind myself, because I'm going to keep up with how many carbons I have, and I'm going to do that by putting the number of carbons, and I'm going to circle it to keep up with how many I've got. So I start off with 6. Then we're going to claim that glucose, because I think what I'll do now is I'll put, I'm going to draw that. So I'm going to let this be my cell membrane. I'll remind myself of what I did not draw. That it is a phospholipid bilayer, and we'll come and put some more detail in here later, but glucose is going to come into the cell, so I'm going to just draw it again. Now that I know what it is, I don't have to keep calling it anything, and I'm going to remind myself that we still have 6 carbons. Once it's brought into the cell, glucose is going to be substrate level phosphorylated twice, which means we're going to add a phosphate group. That's going to, now glucose already has weak unstable bonds, and you just zapped it with the cattle prod, so it's going to have lots of more instability. So I'm going to draw that I now have this phosphorylated. So a phosphate group was added, and I have to show what did that. So that was at the cost of ATP, forming ADP. So it gave its phosphate group here. Then glucose is going to be phosphorylated again, and it changes its configuration. Now I'm not being exact here, I'm just making the point that we've got this difference now and what the molecule looks like, we still have 6 carbons. We have not done anything about that. No bonds have been broken. We have phosphorylated once, now twice. So this second phosphate was at the cost of an ATP. So we have now a phosphorylated twice glucose. It's ready to be broken. It can now be, it's energetically unstable enough that it can be broken, and we're going to break it in half. So we're going to create 2 3-carbon molecules. So I'm just going to show that now we're going to have 2 3-carbon molecules. The name of one of them, I don't know these names as far as just what they are, but it's going to be a 3-carbon molecule that's the isomer of another 3-carbon molecule. And as weird as it is, this one is usable, this one is not. So this one then gets converted into this isomer of itself. And 3-carbon. So we took the 6, keeping up with them. There's 2 3s. So we're going to, at this point now, we're going to convert this 3-carbon molecule. Now there's a lot of steps. And we're not going into them. You don't have to know them. But there's going to be a process where, through changing this format of the molecule, which is more unstable with weaker bonds, into another molecule that is more stable, we're going to have energy released, and we're going to gain ATP back. So we're going to convert this G3P into a molecule called pyruvate. Why did I give myself no space? So pyruvate, still a 3-carbon molecule, but this more stable molecule is going to create not 1, but 2 ATP through this pathway that we did not go through. And because a bond was broken, we're going to also have to deal with an electron. So 2 electrons, 2 hydrogens. So you're also going to form 1 NADH from the NAD. So there were electrons generated, they were picked up by NAD, forming NADH. And because there's 2 electrons that can sit here and there's 2 hydrogens generated from it, we're going to account for the hydrogen ion that's just free. So that's going to repeat itself up here for this other. So have a pyruvate formed again. We have not lost carbon. But in the restructuring of this G3P into pyruvate, these weak unstable bonds become more strong and stable, and so that energy is used to substrate level create the 2 ATP, 2 for each, and you're going to have 1 NADH. So there were electrons formed in this reconfirmation, and so you've got an electron carrier there. Okay? So this is inside the cell. All of this right here is in the cytoplasm. And I want, but I do want orange. Do I have one? Isn't it crazy how we get like stuff something that we want? So I found my orange highlighter. So I'm going to remind myself that all of this right in through here all of this from the cell membrane in. This is the cytoplasm. And this process of taking glucose, which I think I am just going to remind myself that it's glucose, phosphorylated twice, snapped in half. Now I have 3 carbon pieces. I restructure it to make it to where it's less uncomfortable with itself. It's more stable, forming pyruvate. And I'm going to generate ATP, not a lot, substrate level, you don't make a bunch, and a couple of electron carriers, and now I have pyruvate. This whole process right through here is glycolysis. So I'm going to come up here and I'm going to write glycolysis. So glycolysis, we can then later down here figure out what we used, what we gained, all of that down in here. We're drawing. Okay. So, we're still in the cytoplasm. Me and my orange highlighter. Alright, still in the cytoplasm. The pyruvates, in order to be oxidized, broken down, have to move into a mitochondrion. So now that's what I'm going to draw. Now, I'm going to make it big because I got a lot of stuff I got to do in there. So I'm going to draw this scary, ginormous part of the mitochondrion, knowing that it has this inner membrane, all about that endosymbiotic theory, where the double membrane means engulfed, has its own DNA, has its own ribosomes, replicates on its own schedule, all of that evidence. So this now, I know what this is, right? So I'm going to write under here, this is the mitochondria. And I'm just going to make it plural because it would be where it would go into a bunch of them. So this pyruvate enters into this mitochondrion through a pyruvate channel transport protein in the membrane. Because remember, I'm not drawing it, but this outer membrane is a phospholipid bilayer. This inner membrane is also a phospholipid bilayer. So I just don't want to get myself to where I'm thinking because I drew one line that I'm just saying something. They're both these phospholipid bilayers. So I'm going to draw this channel protein right here, and I'll label it what it is. So this is pyruvate, lost my place there, pyruvate channel, transport protein. So that's in the bilayer. And they're going to be all in there. So I'm going to draw just several to let myself know that they're all throughout there. When the conditions are correct, you're going to be able to have this pyruvate molecule move through that transport protein. So let this one go through here. So now it's moved into the mitochondrion for the rest of what's going to happen. It's in this in-between inter-membrane space. So I'll write all this in just a second, and I'm going to color and all that. But there will be another transport protein in this inner membrane. There's going to be whole bunches of them. And this is an interesting protein, this transport protein. So write what it is. This is your pyruvate, again, transport protein, but it's a SIM-port type of protein. So this is a SIM-port, which means together transport protein. And what it will do is it will have hydrogen ion, will move with its gradient so that tells you right away that hydrogen's high gradient here, low here, because it's going to move with its gradient, and it grabs the pyruvate moves it through with it, but that's against the gradient. So that's a pretty interesting use of, as pyruvate comes here, this now is going to be where pyruvate's low. It would not naturally move in here where there's lots of pyruvate. So you bring it with the hydrogen that will move naturally through. So I'm going to show it this way, where I'm going to rewrite pyruvate so I know it came into here. So here's pyruvate. I'll remind myself it's 3 carbons. It's going to come through this transport protein. So pyruvate comes in. It is brought in with a hydrogen ion. So I'll do the same thing here. I'll put that pyruvate came through this outer membrane channel transport protein, 3 carbons, and it's going to be driven in here into this inner space because hydrogen ion will come with it. So now we have pyruvate in this middle space. This is called the matrix. This space in between the 2 membranes is called the intermembrane space. Out here, of course, is our cytoplasm. So I'm going to keep giving myself that color so that I can associate what is where. So I'm going to stop this for a second. I'm going to draw, you draw. I'm going to do this shading. I'm going to go get a yellow. You could use a highlighter, but I'm going to go get a yellow colored pencil. And I'm going to make this very pale. And I'm going to make this intermembrane space darker. Then I'm going to add a lot of these positive charges because instead of making a bunch of hydrogen ions, it's easier just to show them as the protons that are the positive charge they are. Okay, so I'm going to pause it, and I'm going to go color. You go color. Okay, so there we are. This part right through here, so this is yellow, you can see, so it's barely yellow, and then this is darker yellow just to show the gradient, where there's more hydrogen ions in this intermembrane space. The concentration's higher than in here. So I'm now going to deal with the pyruvate that we were able to bring into the matrix, and I'll spend a little bit of time once I've got what I want here to just show that there's quite a bit of pyruvate in there. So this pyruvate is now going to, got 3 carbons, right? We're going to go through pyruvate oxidation, and we're going to reduce the number of carbons in the pyruvates to 2. So we're going to lose a carbon. So I'm going to just give myself this pyruvate right here. And I'm going to show that it's going to lose a carbon. So we're going to break a carbon off, and carbon dioxide's going to be formed. When we broke that bond, there were electrons that were created, lost. So we're going to give those to NAD, forming NADH, and a hydrogen ion. Then this 3 carbon now becomes a 2 carbon molecule called acetyl. And we'll give the acetyl group, we'll add it to a CoA. We've talked about that before, coenzyme. And we'll have acetyl-CoA, and it's going to go to the Krebs cycle. So I'm just going to repeat this down here. So I've got the pyruvate, 3 carbons. We're going to break off 1 carbon, which will be carbon dioxide. You'll have, from breaking that bond, you'll have electrons. So NAD will be reduced to carry those electrons. You'll have hydrogen ions created. Pyruvate then becomes acetyl, which is a 2 carbon. We add the CoA to it, and that's going to go off to Krebs. Okay? So that's this set. And it's not short. Now I'm going to come over here, and I'm going to create the rest of the mitochondria. So I think that's what I'm going to do. I'm going to stop this, and all I'm going to do is I'm going to draw the rest of this mitochondrion. I'm going to put this inner membrane in there. I'm going to do my coloring. I'm going to create my hydrogen ion gradient of higher in this inner membrane space, lower in this matrix, leaving a great big place to draw in Krebs cycle. And then in this inner membrane, don't draw it over here. I'll do mine and I'll show you what I'm talking about. Okay, so this is what I was talking about. I'm going to go ahead and I'm going to put the other side of this one mitochondrion in here. But I want to be able to show the electron transport chain in detail in the membrane blown up. So I don't want to have to draw on top of what I've already drawn. So I'm just going to leave this for later, and I'm going to fill that in. So now I am going to stop it. I'm going to do dark yellow, light yellow, lots of hydrogen ions in this inner membrane space, and then I'll come back. So you do the same thing. Alright, so just in my yellow over here, saving this space for my electron transport chain to put it in this inner membrane. I added the detail of, it's not surprising, but that's called the outer membrane, that's called the inner membrane. And then I tried to write inner membrane space here, but my pen didn't want to. And then now I've got quite a bit of room here to show what happens to the acetyl-CoA. And this is going to be where Krebs cycle is. Also called tricarboxylic acid cycle, also called citric acid cycle. So we're going to start off with, and instead of trying to draw the two, I'm going to now just look at one acetyl-CoA, knowing that we had two. So, coming from where we were before, we went through pyruvate oxidation, we snapped off the carbon dioxide, we created acetyl, we added CoA, so we have, acetyl-CoA, reminding ourselves that it's two carbons. As it enters this whole Krebs cycle, which when it says cycle, just remember that whatever you start with, that you end up with it again. So it just regenerates. We'll go through it. This is not about learning. This is about drawing. So you're going to lose the CoA. It's going to go away. Acetyl now is going to be added to a molecule, and I'm going to try to maybe write those in red. We'll see. To a 4-carbon molecule. I'm going to leave room here to write, and this will make a 6-carbon. Sounds like we're going backwards, because we went from 6 to 3 to 2, and now we're stuck with 6 again. But these are just part of this cycle. All we're really breaking down is this right here. Then there's going to be just a conformational change. Still, say it's still 6-carbon. Then there'll be breaking off of a carbon. So this is going to be between here and here. There'll be a generation of carbon dioxide, which it just diffuses on out of the mitochondria because it can go right through those membranes. Because I broke a bond, I'm going to have electrons I have to deal with. So I'll have NAD reduced to NADH. There'll be hydrogen ion formed there. Then this 5-carbon is going to be metabolized, oxidized into a 4-carbon molecule. So again, I'm going to lose the carbon dioxide. Just diffuses on out. And I had bonds broken, right? So I'll have NAD reduced to NADH. So let's draw it from here because my pen's doing better that way. And there's going to be a creation of, and I was going to put it there, but it's not wanting to work, so I'll do it here. GTP that's going to be formed, which will then pretty much quickly be turned into ATP because they're both those nucleosides and it's easy to swap this one out. So we've gone from a 2-carbon added to a 4-carbon to make a 6-carbon molecule that was rearranged, a carbon removed, another carbon removed. Then you're going to have some just conformational changes that you're going to go through. So we're going to go from 5-carbon 4 carbon to 4 carbon, and then more conformational changes to come back to this original one. As these conformational changes occur, there will be bonds broken, so we're going to meet a new electron carrier called FAD, and it's going to be reduced to FADH2. So this is just a different bus, and here we'll have more electrons that will be generated and picked up by NAD. So as we come back and we learn this, we'll come put the names in here. I'll use my red pen, shows up better. But this is called the Krebs cycle. So at this point, let's think about what's happened to our carbons. We had 6, we snapped it into 2 3s. So since it's the same thing for each one, I'm only going to follow the one. We had this 3 carbon molecule formed into pyruvate, going through a series of just energy changes. This is more stable. Still 3 carbons. So we haven't lost any carbons yet. We've got 2 3 carbons. Pyruvate comes into the mitochondrion. Then we start losing carbons. So pyruvate is converted to acetyl with the loss of a carbon. So we've lost 2 carbons there. Now we're to 4. So there's acetyl-CoA goes through. The second acetyl-CoA goes through. So we went from 2 carbons, and then here we lost 1, and we lost the other. So we have completely, after Krebs turns twice, for each glucose, we have no more carbons. Everything is done when it comes to breaking, oxidizing glucose. There is no more glucose. It is all converted into carbon dioxide. But we have to deal with the electrons on the bus. So we're going to now come over here and draw that factory, the electron transport chain that we drew in isolation there. We're going to put that in the membrane over here. So I'm going to put in the first protein that we had talked about, where this is the parking space right down here. NADH parks here, gives up the electrons. So this is called, believe it or not, complex 1. So just like we did before, we're going to remind ourselves this is in the membrane. You have this odd little protein, this little complex that's 2, but it's not really fully associated as far as it's not a pump. It's not transmembrane. The next complex or part of it is, believe it or not, 3. We're on a roll. Followed finally by 4. And then we have my favorite molecule in the whole world. It looks like a revolving door at a fancy hotel. So it's going to have this revolving piece like that, and this what's called a catalytic knob that's on this matrix side. And that's going to be one electron transport chain. So this is the electron transport chain. And all of this enfolding of this membrane gives you more surface area And so you can have gobs of these. So I'm just going to turn my paper, and I'm just going to do some simple, this is 1, this is 2, this is 3, this is 4. That's ATP synthase. Draw a couple more. There's 1, there's 2, there's 3, there's 4, some ATP synthase. Draw a few more. 1, 2, 3, 4, ATP synthase. That's enough. I get the point. That the membrane is loaded with these. So, the way that this works is we're going to have a parking space for NAD. So, NAD is going to come here, just like we drew before. So, NADH, because it has a full bus, it's carrying electrons, it'll park, drop its electrons off, become NAD plus, and then it's going to go back, and it can pick up electrons anywhere that we needed to have it as this continues to go. Then we said we had a new electron carrier buddy, a new bus. It has its own parking space at 2. So FADH2 parks at 2. It empties its bus, and it's now empty and can go back over here and pick up more electrons. So we had the electrons before. We showed them as being dropped off. They'll move through the complex, and the electrons then will be given to water, and we're going to form water. Now we haven't explained anything. We've just drawn. So the only thing I have left to do is to draw in this gradient in this inner membrane strip space. So as we learn about this, we're going to show the pathway of all of these NADHs coming here, and all of the empty buses, the NADs going back. So we'll add here that we converted from NAD to NADH. So this is not about learning. We'll have details that we'll add as we go, all into this space down here that we saved. But we're through with our drawing, so I'm pretty shocked. I thought that would take longer. Not meant for you to be able to go, oh, I know every bit of that now. It's one drawing for reference, so when we start doing our information, we can reference a detailed drawing. That's all it's about. Okay. There will be no video. Nobody else does this. There's a good reason. Pretty crazy, but it is the best reference to always come back to do. Color's important. Don't forget color. You're amazing.